Okay, next in line is our fourth oral presentation for today. I would like to welcome Miss Atieya Abd Hadi. Let me introduce about her. She is successfully obtained her Bachelor of Science in Industrial Biology and Master of Science in Biotechnology in University Technology Malaysia. With the desire to venture into interdisciplinary research, she is currently pursuing her PhD at the Department of Biosciences under the supervision of a Associate Prof. TSCHM Dr. Ni Ahmad Nizam, starting from 2019. Currently, she is working on the antibacterial activity of the green silver nanoparticles towards resistant bacteria. Her research interest is green synthesis, silver nanoparticles, antibacterial assays, and nanoinformatics. So her title for today is Insilico interaction analysis of silver nanoparticles towards opportunistic pathogens protein using stage 5.0. As usual, you have 10 minutes for presentation and 5 minutes for Q&A session. Miss Atieya, please welcome.
Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Does my slide appear?
Yes, clear.
Okay. So I can start now. My name is Atieya Abd Hadi. Thank you to the moderator. So my title for today is Insilico interaction analysis of silver nanoparticles towards bacterial protein using stage 5.0. So basically, since in the morning we have heard so much about silver nanoparticles, so today I will focus on the antibacterial interactions. So we know that silver nanoparticles have outstanding credibility in antibacterial activities. So here I highlight the three important key points of my presentation. Firstly is the interaction analysis. So here I will share a bit about stage 5.0, which is a screening software I use to see the interaction of silver nanoparticles. So secondly is the silver nanoparticle itself as the compound. And thirdly is the bacterial protein, which is the target of the whole antibacterial activity. So just briefly, when we say about Insilico, so actually it defines a really big scope. So the name Insilico actually comes from when we do computational approach on silicon. So silicon is actually a chip. So it defines the chip we use in the computational. So simply we can say that in experimentations, methods or predictions, we use a computational approach. So why we use this computational approach? First, because it can produce high simulation accuracy, and also when we want to discover drugs, and also for example that we want to design any molecular compounds. So actually when we use this method of in silico, we can save a lot of time by screening the compound and also without risking any tedious lab work. And also when we do in silico works, it is beneficial when at times like this where we cannot enter the lab work, and when we want to do lab work, we need to choose which lab work that we want to do. So actually there are lots of benefits when you use in silico works. And as I mentioned here in this diagram, we can say that there are lots of scientific literatures, millions of articles, and now they are put into the database. So actually we can get higher accuracy results based on the data mining from the results. And also we can predict what diseases, what compounds, what are the cells and the functions involved. So actually I can say that in silico is the best friend of experimental works. So my research problem, so basically I've started the work with biosynthesis of silver nanoparticles. Then I optimize the silver nanoparticle production using Persicaria odorata as mentioned from the technique. And I did in vitro antibacterial assay using these three techniques, these diffusion techniques, where we get zone of inhibition which is quite large, and it is comparable to antibiotics. And when we test with minimum inhibition concentration and minimum bactericidal concentration, we got good results from our biosynthesized silver nanoparticles. However, the gap of the study is the bactericidal activity of the biosynthesized silver nanoparticles is not yet extensively studied towards resistant bacteria. So there comes the interest where we delve into the resistant bacteria. Just a simple introduction why I want to focus on resistant bacteria. This is because as we all know, when bacteria, virus, fungi and parasites, they no longer respond to medicine, it will make infection harder. So when infection is harder to treat, uh, regardless of any designs of antibacterial agents, we cannot cure because the bacteria has become powerful than the compound, than the antibacterial agents itself. So I would like to elucidate the antibacterial mechanism towards the resistant bacteria using this overlap approach in vitro, which I already got the results, and also the Insilico approach. So this is the rough framework of it. Okay, when we want to do in silico works, we need to have a target and also a compound. So the compound is biosynthesis nanoparticles. So actually, there is also a computational work there, which involve computational chemistry, as explained by Dr. Fazira earlier. So I would like to focus on this, which is the target where we want to screen the target binding protein. So in this particular area, I want to choose two model organism, which is methicillin resistant Staphylococcus aureus (MRSA) and also Pseudomonas aeruginosa. So MRSA represents gram-positive bacteria. P. aeruginosa represents gram-negative bacteria. So actually there is a problem when we want to choose which bacterial protein. There are too many bacterial proteins and we know there are rough mechanisms involved. But however, which bacterial proteins that we want to choose? Is it based on the literature or how? So actually this is where the screening using in silico tools comes in handy. So actually this is the first part.
Sorry, Miss Atieya, to
Yes,
interrupt. There is another 4 minutes to
okay.
finish the speech.
So we will use Teach 5.0. So the objective is to study the interaction between AgNP towards the two chosen bacterial proteins, two chosen bacteria, and to deduce the bacterial mechanism. So this is the interface of the stitch. So we can, it is very advantageous because it is an open source and it is free, so everyone can use, and also it has constantly expanding database. So for example, like this, we just enter the compound input using chemical structures and everything, choose the organism that you want. It also has human proteins also, and obtain the network. So we can see that there are various databases incorporated in this network. So the results and discussion. So this is for P. aeruginosa. We can see that the network is we choose the confidence score of 0.8 above, where we see the compound target interaction. So we can see that it targets the multi-drug antibiotic resistant efflux pump system, which is at the outer membrane protein, and also attacks the inner cell cytoplasm's protein, the intrinsic proteins. So this one is for the MRSA. So basically we can see that majority of the uh, binding of silver nanoparticles is towards the intrinsic protein, which it disrupts the cell homeostasis. Okay, so basically, when we do the network analysis, so we outline it with the antibacterial mechanism. So actually, there are around 7. So based on the results, we can say that the antibacterial mechanisms is different because there is difference in the cell wall of the gram-negative bacteria and the gram-positive. So basically gram-negative, it interacts with the efflux pump, which is at the protein membrane, at the membrane of the protein. So for the MRSA, it interacts inside. So this is the conclusion. So we can see that the functional protein network is clearly derived. For gram-positive bacteria, we can see it causes major intrinsic destruction. So that's why the MRSA is killed by the AgNP. And also, I can say that the gram-negative bacteria is also obstructed by the silver nanoparticles strongly. So actually this result can be further used in molecular docking and dynamic simulation to actually visualize the interaction. So this is my acknowledgement to my highly motivated and ever supportive supervisors and co-supervisors and the grant and also the sponsorship GPA Malaysia. So that's all. Thank you.
Okay, thank you, Miss Atieya, for your wonderful sharing. So right now we will move on to the Q&A session for about 5 minutes. Okay, Miss Atieya, we have one question. Okay, let me read for you.
Okay.
Does Stitch database also include progressively mutating bacteria?
Yes, for example, MRSA, there are, when I see that the input organism, there are a lots of Staphylococcus mutants, MU50, PA01. So actually, if the database, because the database is continuously expanding, so I expect that more mutating bacteria will be included since from the literature we can say that since 2020, 2021, many are shifting its what we say antibacterial works towards the resistant bacteria. Since we know that the COVID-19 is also accelerating the
Yeah, yeah.
antibiotic resistant problem.
Okay, for another question, do you plan to try other computational method and how do you compare Stitch 5.0 finding with experimental findings?
Oh, actually, I decided that I want to compare with the cytoscape, but I think need some time to explore. The cytoscape really how I say does it really can give the results that I want. So when we the second question is compare Stitch with the finding. Yes, actually uh, when we compare the finding, actually it complements. For example, that when we use Stitch, it says that it attacks the nitrogen oxygenase inhibitor something. So we can plan to choose which antibacterial experiments to verify that. I was thinking of it can help us to choose which experimental works that we can do to strengthen the results from the stage.
Okay, there's another question more. Okay, what makes AgNP different from current antibiotics used to cater the MRSA and PA infection? Is there any difference in terms of the mechanism between AgNP and antibiotics?
Okay, I didn't include this one so because I know the the presentation will be longer. Actually, I've compared the antibiotic of chloramphenicol. I did the network analysis with the same MRSA and also P. aeruginosa. So actually, we can see that the antibiotics work by binding towards lipid protein. So when we compare with the silver nanoparticles, I think it is quite how I say intelligent ke? Because actually it targets two things. Silver nanoparticles, first it will bind to the pore membrane. When it binds and actually it makes the membrane haywire, so more nanoparticles will come into the cell. So actually that one is a really excellent work of the nanoparticles. So for example, antibiotics, uh, it is really dose dependent, right? We need to
Yes.
increase the dose at a higher dose when it doesn't become efficient. Why? Because it doesn't able to penetrate inside the cell, because mostly this antibiotic will work inside the cell. It works wonders inside the cell, but it needs to have like something to bind specifically towards the cell wall actually. I think actually both works, but I think the synergistic effect is better. That,
Okay. Okay,
yes.
Miss Atieya, thank you. Do you
Thank
have
you.
any other question? Okay, thank you, Miss Atieya.